Hi everyone, my name is Alexandra and welcome to this video on experimental study design. Experimental studies are the second major category of research study designs. And unlike observational studies, which we've been talking about up until this point, in experimental studies, the researcher directly manipulates the independent variable. So for example, let's say we were interested in learning about the relationship between radiation exposure and tumor development. We could take a group of mice and assign half of them to a group that is going to receive radiation. And then we would leave the other half unexposed to radiation, and that's going to serve as the control group, which serves as a comparison group for the mice that are receiving the radiation. Now, let's say that after doing these studies, we notice that the mice in the radiation group are developing tumors, and the mice in the control group are not developing tumors. So based on our study results, we can say that radiation exposure causes the development of tumors. Notice that this is the first time in this video series that I've said a study can show that an exposure causes an outcome. This is because experimental study designs are the only type of study that can directly assess that causal relationship. Let's talk about why that is. So in our study example from before, we were hypothesizing that radiation exposure causes tumors. But we can come up with a list of other things that might cause tumors. So, for example, maybe the age of the mouse matters, maybe the sex of the mouse matters, maybe the genetic background of the mouse matters. So how do we know for sure that it's the radiation that's causing the tumor and not these other factors? And that's the purpose of our control group. So in our group that is not receiving radiation, we can make sure that we're including mice that are the same age as those that are receiving radiation. We can make sure that there's similar proportions of male and female mice in both groups. We can ensure that they're eating the same kind of food and living in similar environments. And we can even make sure that they have the same exact genetic background. And what controlling for all of those other variables does is it allows us to be more certain that the only difference between those two groups is the fact that one is receiving radiation and the other is not. Therefore, if we see a difference in outcomes, for example, if one group is developing tumors and the other is not, the reason must be because of the exposure. So the reason that experimental studies can assess causality is that the researcher has a lot more control over all of the other variables compared to an observational study. Experiments where an exposure is hypothesized to be harmful are unethical to do in humans, so we can't take a group of humans and expose half of them to radiation and see if they develop tumors. However, if the exposure is hypothesized to be helpful, we can do that study in humans, and that's called a clinical trial. In a clinical trial, a newly developed treatment is being compared either against the currently available treatment, or the treatment is being compared to a placebo, which is a substance that mimics the treatment but doesn't actually have any therapeutic effect. So in a clinical trial, you might recruit a group of people with the disease that you're interested in treating, and then you would assign half of them to the treatment group and half of them to the placebo group. And then you would assess the outcomes and see if the patients in your treatment group become cured of the disease more often than the patients in the placebo group. Let's talk about the characteristics of a good clinical trial. Clinical trials provide the highest level of evidence when they are randomized, controlled, and blinded. Randomization refers to how participants are grouped into either the placebo or the treatment group. You want to randomize this process so that you aren't introducing bias or systematic error into your experiment. So imagine that you're running an experiment and you want to recruit 100 participants. If you take the first 50 participants you recruit and put them in your treatment group, and the last 50 participants you recruit and put them in your placebo group, so in a way that is non-random, you don't know that there isn't some difference between the first 50 people and the last 50 people. For example, the first 50 people might be very interested in your trial and very much want your treatment to work, but the last 50 people might be less interested in your trial. So you've now introduced this extra variable that you haven't controlled for. Randomizing how study participants are assigned to the treatment and control group helps ensure that both of your groups are as representative of the general population as possible. Controlled refers to the presence of a comparison group that is not receiving the treatment. An important aspect of a well-controlled study is you want your control group and your treatment group to be as similar as possible in all other variables other than the fact that one is receiving the treatment and one is not. So for example, you wouldn't want the average age of your treatment group to be 60 and the average age of your control group to be 30, because if you found a difference in outcomes between your treatment and control group, you wouldn't know if that's because of the treatment or because of the age difference. When you read a clinical study paper, you'll often see a table comparing the demographic information of the treatment and control groups, and that's how the researchers are demonstrating that they've done their best to control for other variables other than their independent variable. Now you might be wondering, how can you both randomly assign participants into groups and also make sure that the groups are similar to one another? And a common way to do that is called blocked randomization, where your participants are first grouped by variables you would like to control for, and then randomly assigned to your study groups in equal amounts. So for example, you could group your participants into young and old groups, and then randomly assign half of the young participants into the treatment group and the control group, and then randomly assign half of the old group into the treatment group and the control group. And in that way, you've randomly assigned equal numbers of young and old patients into both treatment groups. Now, in a blinded study, participants that are in the study don't know which treatment group they've been assigned to. Usually that's done by ensuring that your placebo group still thinks that they're participating in some kind of treatment. So they might take a pill, but the pill is just a sugar pill and doesn't actually have any of the therapeutic compound in it. In a double-blinded study, neither the study participants nor the researchers that are running the study know who is assigned to what group. And in a triple-blinded study, participants, researchers, and also the people who are analyzing the data at the end of the study aren't aware of the group assignments. This helps reduce certain psychological variables that can enter into a study. So for example, if you are a participant and you know you're part of the treatment group, you might be more likely to report an improvement in symptoms than if you thought you were in the placebo group. Next, I'll discuss crossover studies, which are a special type of clinical trial typically used to compare two different treatments. In a crossover study, half of the participants receive treatment A first, and then there is a washout period where participants receive no treatment and the effects of treatment A fully wear off, and then those participants receive treatment B. Now, to ensure you're controlling for the potential effect of the order that the treatments are taken in, you take the other half of your participants and give them treatment B first, and then after the washout period, they receive treatment A. And what this does is it allows the patients to serve as their own controls. Because all patients are at some point receiving treatment A and treatment B, you can compare the effects of treatment A and treatment B in identical populations. And this helps reduce confounding bias, which is effects that are caused by variables other than your independent variable. However, this design isn't always possible for all research questions, so you can imagine if you expect treatment A or treatment B or both to cure the disease that you're studying, you won't be able to complete the second half of the study and your study will be meaningless. This study design is really best used for chronic diseases. So for example, you could compare two different blood pressure medications, but you probably couldn't compare two different antibiotics. Okay, the last thing I want to talk about is the phases of drug trials. In phase 1, the goal is really to determine, is the drug even safe to use in humans? So only a small sample of participants are recruited, and then researchers assess the toxicity, pharmacokinetics, and pharmacodynamics of the drug. If the drug is determined to be safe, it can move on to phase 2, where a larger sample of patients is recruited, and the researchers assess whether the drug actually has an effect on the disease and what the best dose for the drug is. They'll also continue looking out for any side effects that might make the drug dangerous to use in humans. If the drug seems to be effective and continues to be safe, it can move on to phase 3, which is a much more robust study where hundreds or thousands of patients across multiple centers are recruited, and typically the drug at this point is being compared to the current standard of treatment. So is it better than what's currently available? If the drug continues to be safe and effective in a larger number of people, the pharmaceutical company can apply for something called the New Drug Application. And at this point, the company is requesting approval from the Food and Drug Administration to be able to sell the drug to patients. If the drug is approved by the FDA, it moves on to phase 4. In this phase, the drug is now available for patients that aren't directly enrolled in a clinical trial to use, but the pharmaceutical company must continue to watch out for any long-term or rare side effects that might not have been noticed in the phase 3 clinical trial. Let's finish by highlighting the main unique points that can help you identify and distinguish experimental studies on test day. The first thing to do is identify that a study is an experimental study by noticing that, unlike observational studies, patients are being assigned by the researcher into an experimental or control group. So the researcher is deciding what kind of treatment a patient gets. If participants receive both treatments over the course of a study, and there's a washout period described, that's likely a crossover study. For differentiating drug trial phases, pay attention to how many people are enrolled and what kind of question is being asked.